Okay, we begin the second part. Important terms. Before we discuss this, the laws of segregation, the law of independent assortment, and the law of dominance, we need to know certain terms. And the first of which is gene and allele. Gene is the character. Gene or character is any inheritable feature or character, whereas allele is the variant form or the alternative forms of a character. Allele actually is the shortened form of allelomorph, which actually means the other form. For example, let me give you the example of hair color. Black and blonde. Or blonde is the recessive version, whereas black is the dominant one. So these are black and white, two colors, blonde and black, two colors. Both resemble the one character, that's the color of hair. But there are two, versions, black and the blonde. So the variants are allele, whereas the whole character that we talk about is called the gene. The next one is dominant and recessive. Dominant trait is the one which actually expresses itself or manifests on the organism. It masks or it covers up the other traits that are there. So dominant one actually expresses itself in the organism, in its phenotype. Whereas recessive is the trait which is masked by the dominant trait and it remains hidden in the gene. Phenotype and genotype. Phenotype is the trait which appears on the organism. So that is the dominant one actually appears in the phenotype. And genotype is the genetic combination. Move on to the next one. Homozygous and heterozygous. When the traits are similar, whether it be capital T, or small t, if they're same, it's called homozygous. When it is dissimilar, then it's called heterozygous. Capital T and a small t, that's the heterozygous. The next one, F1, F2, and P. I'll start with P. P is, the parents is denoted by the capital letter P. Okay? And after the parents are decided, when they are crossed, the first generation that is formed is called the first filial generation. And the second filial generation, in order to achieve the second filial generation, we need to cross the first filial generation. So you can think of parent being the grandparent of the second filial generation. So not to confuse. Okay? F2 is the grandchildren of P, so to speak. Okay, the next one. Locus or loci. Loci is a particular place of a trait on a chromosome. I'm going to take up the example of the X and the Y chromosome of the human sex chromosomes. They are the chromosomes. These two chromosomes determine the sex or the gender of humans. Okay. If you look at the X chromosome, there is a place here where a trait, a character is present, a gene is present, okay, or a trait of a gene is present, known as colorblind. And it's present on the X chromosome, but on the Y chromosome, that place does not exist at all, because the Y chromosome is much smaller than the X chromosome. So we can say colorblind trait has its locus on the X chromosome. So locus is the actual place of the gene of a trait on the chromosome. Okay? And homologous chromosomes. Homologous chromosomes have corresponding alleles. Look at this example. There's a pair of chromosomes with similar colored genes. Let's say the red one stands for, this is orange. It stands for, let's say, the features of the toes. And this pink one stands for the features of, let's say, the eyes. Chinky eyes, big wide eyes, okay? Big eyes. This one is toes. It stands for toes. So these two chromosomes have similar genes. Genes for toes, genes for eyes. Genes for toes, genes for eyes. And similar colors stand for corresponding genes. Whereas in the case of non-homologous chromosomes, we find that the red does not have a corresponding red on the other side. So if this stands for toes, this may stand for ears. So such kind of chromosomes we call non-homologous chromosomes, where the genes are not corresponding to each other. I mean, they are not similar. They don't stand for the same character. The next one. The law of dominance, having having gone through all those terms, now we are ready to tackle law of dominance. The first one is law of dominance, which states when contrasting pairs of alleles of gene is present, the dominant trait masks the recessive and expresses itself in F1. Now this can be explained by this simple cross between a pure tall and a dwarf plant. Now a dwarf plant is given by small t, and a tall plant is given by a capital T. So these are the parents, and we call this as a pure tall because there can be another tall which is heterozygous tall, capital T and a small t. That would be a heterozygous tall. But here is a pure tall plant which is crossed with a dwarf. The genes of the pure tall plant are here, capital T and capital T. And genes of the gametes of the dwarf plant is given on this side, small t, small t. When you arrange it now, we always write the capital T's first and the small t afterwards. So here, you get a capital T from here, and this T goes here. This capital T drops down here, and this small t goes here. So we have capital T and small t, capital T and small t. All of the boxes have the same genetic combination. Now when Gregor Mendel crossed a tall plant with a dwarf, he always found that the first filial generation, all the plants were tall. He did not know, then in his time, the genes were not discovered at all. We did not know that all of this feature was actually controlled by the genes that are present in the chromosome. The next. Phenotype, all plants are tall. Okay. The second one, a law of segregation. It states that when contrasting pairs of alleles of a gene is present, during gamete formation, they separate completely. So now we're talking about meiosis, okay? Meiosis and the meiotic division, which actually creates the gametes, which I shall deal in another video. Okay? For the time being, just understand that when gametes are formed, these traits, this capital T and small t, they separate completely. Tall and a dwarf, they don't mix to form a gene which contains a medium height pair. So it's not mixed. The genes are pure, and it's also known as the purity of gametes. So it's well explained by this. When you cross the F1, the first filial generation, if you cross it, remember back in the law of dominance, we found that the first filial generation was capital T and a capital small t. All of those four boxes have the same combination. Now we are taking these as the parent and then crossing it, and then in F2 we get this set of genetic combination. So one set of gametes we arrange on the top, and the other one vertical as this. And the capital T comes here, and this capital T goes in here. This capital T goes in here, small t comes down. Here again, the capital T drops down here, small t here, and small t here this small t here. Now what this tells us is, phenotypically, or the phenotype, we have 3 tall plants. Because tall, this is a heterozygous tall, and here is a pure tall. And then we have 1 dwarf. So initially we started off with the crossing of this and this as parents, pure tall with a dwarf. And then we ended up in F1 getting all these heterozygous tall. Now the law of segregation is proven by the fact that this trait has again reappeared in this F2 generation. The next one, the third law of inheritance is law of independent assortment. It states that during gamete formation, traits, alleles of different characters randomly rearrange themselves and do not influence each other. In order to explain this, we need to take on a dihybrid cross. Okay? We were dealing with monohybrid cross till now, where we were considering only 1 character. Okay? Now we are taking 2 characters and crossing these and trying to find out what are the genetic combinations that we get. So the different types of gametes that you get on while crossing this F1 generation will be dealt on another video. Okay? For now, just understand that when you go to the F2 generation, you get a ratio, a phenotypic ratio of 9:3:3:1, meaning there are 9 of these, 3 of these, 3 of these, and 1. Okay? So this actually tells us that parental characters are formed and new combinations are also formed, which was not there in the parent at all. So this proves that during the formation of gametes, all these traits, they assort themselves not just on 1 chromosome, but they just assort themselves in different chromosomes randomly. They pick any of the 2 available chromosomes. So I hope this clears, I hope you understand the laws of independent assortment, all these 3 laws. But I'm going to pick up problems based on this on next video.